So currently in the industry, we use these things called manual assays, which, for example, if you want to test how strong a mouse is after some experiment, you hold out this metal mesh, and the mouse literally hangs on to the mesh. And then you time how long they can hang on for. And based on this measurement, you say, okay, I guess that's how strong they are. Cool, moving on. So now imagine doing this for thousands of mice. And somebody is just sitting there waiting for the mouse to lose their grip. So those are the kinds of experiments that are being done right now. It's very, very manual, very tedious. And what we're providing here is a slightly different method. So it's in terms of the Venn diagram, there's absolutely strong overlap. Like we can do object association, see how mice are attracted to various objects, which is very important in neuroscience. We can see how much time they're spending with the objects, how much time they're spending with each other, which is social behaviors, how much time they're spending in like corners of cages or how active they are, right? We have all these metrics we currently analyze that are actually more powerful than a lot of these manual assays.
All right, this week I spoke with Michael Kaca, a co-founder of Olden Labs, a startup that is transforming how animal research is done. Now, when I think about accelerating life science, the first place that my mind goes to are platforms like AlphaFold, which just this year won the Nobel Prize in Chemistry for its ability to take these amino acids and predict the structure of the proteins that are created with them. This is a really important part of the drug discovery process. That said, drug discovery is so much larger than that. According to the FDA, it takes about $2.5 billion and over 8 years to go from developing a new drug to putting it into our hands and in our bodies. And one of many steps along that process involves testing the drugs, testing how well they actually work within systems, their toxicity, and how they work within longer clinical trials. And so a critical part of that are animal research studies, and Olden Labs is working on changing the way that's being done by bringing it up to speed through new platforms like machine learning and a really cool sensor dome that they built. Now, for those of you like me who don't love the idea of animals being harmed in order to develop the drugs that we put into our bodies, we start off our conversation with a deeper dive into the ethics and guidelines that govern animal research today. I hope you'll find this conversation as eye-opening as I did. Let's dive in. All right, Michael Kaca, welcome. So grateful to have you here this morning. So we met a couple of weeks ago at a cool hardware hackers event in San Francisco. Your presentation caught my eye. This podcast is about people and platforms who are accelerating science and science discovery. And you've got a really cool product and company that you're working for, where when we go to the website, the first thing it says is Animal Research 2.0. And you also write further, $36 billion is spent annually on animal studies. So to start off, what are animal studies and how do they fit into the larger life science process?
Yeah, absolutely. First of all, thank you for having me here. Pleasure being here, Nabil, at Discovery Engines. And so at Olden Labs, we are tackling the preclinical trials. And in the bigger scheme of things, when you make a drug, you first develop a fun drug, maybe you knock out some genome, you want to see how things behave, for example, right? So you do all this cool science in the front end, and then after that, you have to actually test it on something before testing it on humans. So that's where the preclinical trials come in, and animal research is a really big part of that. So after animal research, we have the actual clinical trials. That's where you test it on the humans. So that's why it's such an important step. We want to make sure we don't just give people anything for testing. And that's one of the big reasons why over 90% of animal trials or preclinical trials fail.
Interesting. Okay, so animal research, really important part of the life science discovery process. I guess what would we risk if we didn't include animal trials, for example, as part of the life science research process?
I think the most important part of the preclinical trial stage is the safeguarding, right? You don't want to give people just anything. I mean, just because something works in a simulation or in a petri dish or in a math equation, it does not mean it's going to work on a human being. In fact, it will probably do more damage than good, right? I mean, think of the human body, we are an extremely complex organism. right? I mean, for a good analogy, if you want to test, let's say, a CPU of a computer, you don't just take the CPU and then plug it into a bunch of wires and see how it behaves. You want to have it be fully integrated in a system, right? Because you might have different voltage spikes across, I don't know, some IC next to the CPU, or maybe you have some manufacturing defect in the wafer, and all these things add up, and now your CPU is malfunctioning. The body is even more complicated than that, right? So the complexity is something that animal research really tackles, right? Hence, we do it on mammals, very similar to us, like mice, rats, etc.
Interesting. Okay, so there's a measure of complexity that we want to be able to test in before we put, drugs, therapeutics inside of humans. Right now, the other solutions like simulations and so forth don't get us to that necessary level of complexity. So we do it instead with mice, rats, for example. Is that right?
Yeah, exactly. And all these other technologies that are emerging, like they're definitely making a lot of great progress in the last decade or so. Like artificial organs, for example, right? Very cool. Do they work in some sense? Absolutely. But once again, they don't get that full picture, right? That complexity is very hard to actually account for if you just look at one submodule.
Wow. What, like you said, artificial organs. What is an artificial organ?
To be clear, I'm not a biologist, so I'm more on the robotics and the AI side of things. But an artificial organ you can imagine is just, let's say, a kidney or something that acts as a kidney. And you want to essentially model the way that a kidney filters things out, for example, or that it replicates some sort of function that is similar to a kidney itself. And you modify it, you run tests on it, you see how it interacts with different things, right? So you can get that model down to a very, very similar level as your real kidney, but without having the full, system around it. It's hard. It's a very hard problem to solve with high enough accuracy.
Interesting. Yeah, one of the things that reminds me of is in therapy, we often talk about, like, I think one of the critiques of Western medicine is that it's so pinpointed to a specific domain where we don't take into consideration how it fits within a larger system, for example, kidney disease or a heart problem and so forth. And so being able to model things holistically is really important to get to a great end result. So some of these artificial sort of organs, or I've heard of things like organs on a chip, might do a really good job of recreating that kidney, but ultimately it's still being affected by other parts of our body that are important. And right now we don't have a great way of emulating that.
Exactly. And I mean, even look at the recent research that's coming out, our microbiome, like what is it? Well, we're learning a lot about it in recent years, but how does it interact with the rest of the body? Well, there's just so much going on, right? It's like the soup of bacteria. And now I saw some papers coming out recently where basically the brain isn't the only thing responsible for our nervous system, right? It's like, well, now there's a bunch of things they're finding around the microbiome. So it's like, okay, where does it end? Like, what is going on, right? Like biology is hard, and we're still babies trying to figure it out.
Wow. Okay. So I, grew up with animals, love animals. I trust you do as well. The idea, like, I don't love the idea that we are testing on animals. I mean, in general, right? I remember being, happy when we stopped things like cosmetics testing and so forth. We're still doing that in a big way in the life science space. What do you say to folks who like recoil at the idea that we are, putting these, cute little mice and rats in cages and testing on them? How do you respond to that?
Yeah, absolutely. I think it's a very valid concern. I mean, I think ethics should apply to all living organisms, not just humans, right? I mean, I used to own a dog, so I love animals. When I see a spider in the house, I don't kill it, I put it outside, Like, I think life is important, but at the same time, we do need to test things on living beings before we test them on humans, right? So now how do you find this balance of, ethically testing on animals while getting enough data out of them, right? So I think that's why it's important to follow principles such as the 3R principles, right?
So what are the 3 Rs?
Yeah, so we have refine, reduce, and replace.
Okay.
Right, so we'll start with reduce. And this is mostly around the animal research space, is where the 3 Rs are applied. They actually have conferences around it. They had one in South San Francisco here a few months ago. Anyway, so reduce is really to reduce the number of animals being used or to reduce the waste. Right? I mean, if you think about an experiment where, let's say you have 3 mice in a cage and you want to monitor them for 2 weeks or so, right? it's very easy to make a mistake. So what I mean by that is, let's say a lab technician or a scientist checks up on them, every day or every few hours, but now they get sick between days 5 and 8. Okay, they come back on day 8, and now 2 out of 3 of the mice are dead. It's like, well, what happened, right? And it's hard to tell because you put all your eggs in one basket, that one basket being, let's say a single researcher. Okay, so fine. So what happens now? Well, now you have a single mouse in this cage and an experiment that's maybe halfway done, maybe a third, maybe almost done, whatever. But when you have a single mouse left in a cage, you add additional stress to it because mice aren't, essentially, they're not very good at living alone. And that's why single mice cage studies are so problematic, right? And a lot of technologies that try to monitor animals, they're only capable of doing it with one mouse per cage, which adds a whole new variable to the experiment, and really it adulters your results. So long story short, the reduce part is to reduce that waste to make sure that we don't get to that point where animals are dying, they're getting stressed, you have to repeat all these experiments, you're wasting resources, time, you're wasting animals, right? There's a lot of waste. So that's reduce. To refine is to actually make the processes better. So it kind of ties in with that a little bit, right? So for example, you can always make sure that the animals have enough food, water, they're not fighting when they shouldn't be fighting, that they're living a good life, they're not exhibiting any weird behaviors, right? You sometimes want to intervene, sometimes you want to just watch. But regardless, by refining the processes and having better tools, we can learn faster, we can move quicker, and we can make sure the animals have a better life. Great, so that's reduce and refine. Awesome. How about replace? Well, you mentioned things like the organs in the chip, or the computer simulations, things along those lines. Now, I think replace is a very interesting word to use because, like I mentioned earlier, you can't really replace animal studies, right? I think you can augment them or you can supplement them with these alternative methods that we mentioned. But I think what people are trying to say when they say replace is you want to replace testing on 100 animals, for example, to testing on 80 plus 50 of these organs on a chip or whatever.
Interesting. Okay. So reduce, refine, and replace. So these are, it sounds like, ethical guidelines. There are conferences around these. Exactly. Looking forward to hearing more about kind of what your platform does and how it sort of supports those goals as well. Cool. So let's talk about Olden Labs. So what is the origin story of Olden Labs? What inspired its founding?
Absolutely. So one of my co-founders and our CEO, Michael Florea, he did his PhD at Harvard on longevity over many years, and he noticed a very clear pain point, right? I mean, dealing with animals for whatever purposes, especially for longevity, when the experiments are months long, right? In a lot of cases, you have to shovel literally and figuratively a lot of feces, right? It's very manual, it's very laborious, it's very easy to make a mistake. Mistakes can, amalgamate over time. It's a big problem. And when you look at the scope of technology outside of the animal research space, like we have robots that walk around on 2 feet, right? We have ChatGPT, we have all these amazing tools, and it feels like animal research was kind of left behind, like trailing. So that's a big pain point he's experienced firsthand. And obviously, we talk to a lot of people, researchers, scientists, and it's a very common pain point. So a very obvious thing to think about is, why hasn't anybody done this? And the short answer is they've tried. And in fact, a few years ago, I think a decade or two ago, the company that got the closest to it, they were selling systems for about $500K, right? So the barrier to entry with $500K is not a barrier, it's a brick wall, right? And a lot of...
What kind of systems were they selling for $500K?
Imagine smart cages, so fully integrated, like a nice camera system. A lot of them have sold full racks. So you have racks where you put the cages inside, and then all of it was nicely integrated, cameras, system, compute. You analyze the animals, their behaviors, mostly around like mouse tracking technology, right? So a lot of computer vision. And the problem with why that didn't work was not only because the barrier to entry was so high, and a lot of researchers aren't the richest in the world, right? It's a lot easier to sign something for $2,000, $3,000, $4,000 than for something for $500, $600, $700,000. But also because the technology just wasn't what it needed to be, right? Like today, the convergence of AI, cheaper hardware, more talent, and data, it allows us to do what was attempted many years ago with a higher accuracy, precision, and at a much lower cost.
Okay, so longevity researcher at Harvard was working on these studies that took, often months with mice. Noticed that there was that was an arduous process that hadn't been updated for a while. And the only really other kind of competition was this super expensive solution out in the market. And so now, I think with more modern approaches and technologies, it sounds like you're able to bring that price down and make it more accessible for folks. Is that right?
Almost fully right, yes. So the technology that was out there for 500 plus K, they actually died out because it's not a sustainable business model, right? And so a lot of researchers and scientists, facilities that do use some sort of tech is all in-house, right? It's all custom made. It's tedious to bring it up. They have a lot of moving parts. They don't exactly optimize things, right? So it's a mess, right? It's a mess that's going on. So we're trying to set the standard of here's our nice, simple, very affordable hardware package. You simply slide it on top of your cage, put it in your existing racks, and run our software. And just like that, you have data, you have reliability, you have robustness.
Okay, cool. So there's a focus on ease of use as well and integrating with existing things. Okay. And so you've got an interesting perspective on this whole I would say, development or approach. You're coming at it from the hardware perspective. As you mentioned, you've got a co-founder who's more on the biology side of things. So you're a chief science officer focused on data science, and you've got a really interesting career in robotics and in hardware. I think you've done stints at most recently like Cobalt Robotics, Apple X, Google X, I would say, Tesla. How did you get personally involved in this?
That's a great question. And I was coming from robotics. I was doing mostly hardware and more software and a lot of mobile robotics, little security guards that are robots, or you basically try to automate anything that moves, really, right? And I always had a fascination in biology, and that definitely started to be more involved in my life when I'm getting older, noticing like health problems or interesting things that when you look into it, doctors are just like, I don't know. Right? So it's like, okay, well, that's not a good enough answer for me.
Sure.
Right? So then I looked into what's actually being done on the research side, and when you compare that to what's being done on the tech and AI side, it doesn't even compare. Like, it's being absolutely just, the juxtaposition is insane. It's basically a pebble compared to a boulder, right, or Mount Everest. So anyway, so I wanted to get involved somehow as I was still working at Cobalt Robotics at the time. And so Michael Floria, and he reached out to me on LinkedIn. And usually when people reach out to you on LinkedIn saying, I have this great new opportunity, or I'm trying to build this cool thing, the first place my mind goes to is, cool, good luck, I'm busy, right? But then this one person, Michael, he was very, very convincing, and his mission statement actually very resonated with me, right? It's like, look, I'm trying to revolutionize animal research. I'm trying to bring it to the future, and I have all these plans in doing a lot more, right? And when I read that, I was like, okay, A, I can actually help here, right? I can actually really contribute. B, this is something that will really help people, not just companies, organizations, get some money for the shareholders, whatever, right? But you're actually making people have healthier, longer lives if this works well. So, I mean, the impact is just tremendous. And I think as a human being, you always want to leave something behind, right? You want to leave some impact. So that was very inviting. We had a lot of good conversations with him. And at the time, Noah Weber, which was one of the other co-founders that had early conversations with Michael. So we all really got along. We had the same vision going forward. We saw eye to eye. And fast forward now, and we've been working together on this for quite some time, over a year and a half, maybe. It's been long. Time is crazy. Anyway, so we've been working on this together. There are 4 co-founders. We have a team in Europe who helps us with a lot of the web app stuff. An associate professor leads that team, and they help us with also some ML side of things for a lot of the data annotation, actually building the models. And we also have a hardware technician who's helping us actually build all of these components, all of our products, because one of the problems that people think they always want to have is too much demand, right? And that sounds really good on paper until you have to actually execute. With a small team, it's difficult, right? So that's one of the scaling issues I think people don't talk about too much. But yeah.
Okay, interesting. So it sounds like, one, a very convincing and persuasive founder. At the same time, you also have an alignment with the mission or the vision of this. So you mentioned Michael, your co-founder. He was involved in longevity research. I find this a lot in companies, that as you're working on some larger vision, there's something that kind of keeps nagging you, that kind of gets in the way, and sometimes those can feel very sort of like boring or kind of like third-tier issues, and then you realize like, oh, nobody's thinking about this, and actually if we can solve this, not only can I help myself, but there's a whole other category of organizations that are also facing this problem, and oh, by the way, that's a market. So I'm seeing that sort of pattern play out here. I'm curious, is there still, so you mentioned longevity, I think another way of saying it, I think, is aging-related research.
Yes.
Is that still connected to the mission of Olden Labs, or is that Olden Labs' eventual mission, or has it shifted more towards maybe more animal studies specifically?
Definitely. So we're targeting the root cause, which is animal studies, and one of the products of that is longevity. There's also, work with oncology, neuroscience, a bunch of different disciplines in the animal research that use animals for research.
Okay.
Right.
Okay.
So we're really working on building the tools that enable people to do whatever research they need.
I see. Okay. So you're building the platforms to accelerate others.
Yes.
Very cool. Awesome. So we've done a lot of talking about this cool platform. Can we see it?
Yeah, absolutely.
Okay.
So
cool. Well, maybe before we go into the actual demo, can you tell us more broadly speaking, what did you bring today?
Absolutely. So I brought one of our DOMs, which it stands for Digital Online Monitoring Equipment. Very sexy name, I know. And essentially it acts as a smart cage, a very cost-effective, high-quality smart cage that streams your data, and then we have a whole backend cloud system that analyzes it 24/7, right? So imagine you have a cage, and then you simply put this new cage lid on top of it. There's a camera in there, some other sensors, and you get all this data from it. Okay, so that's the dome.
Okay.
I also brought a Wi-Fi box, and that is what we use to connect to the network. So you can imagine you have 1,000 of these domes, right? You don't want to connect every single one to the network. That's a nightmare for IT, for everybody. So then all of these automatically connect to our Wi-Fi box, which is a device gateway from a network perspective. And then this one box connects to the network, right? So you're reducing the number of connections to the network from N to 1.
Okay.
Very simple.
Okay, cool. Go ahead.
Yes, and then the last thing that I brought here is our custom food hopper. And the food hopper is what holds the food and sometimes the water bottle for the mice to eat and drink from. And why do we need a custom food hopper? Because usually they are opaque and the camera can't really see through them, right? So we had to redesign it such that it fits in there nicely, it works with our dome, does not obstruct any vision, and the mice are happy, right? One last thing I'll mention here is that it's important to note that the mice or animals, they do not actually touch the hardware with electronics, right? They're fully enclosed, it's non-invasive, we're watching them and analyzing them with pretty fun tools without actually contaminating anything, so that's never a problem.
Interesting. Okay, so there's an intentional separation between the animals and the kind of hardware electronics. Okay, cool. Well, I'm going to get up and, maybe can you walk us through sort of the design aspects of these three things?
Absolutely.
Okay.
Yeah, so this is the dome right here that I mentioned, this little box, and usually this slides into a rack, right? And we make a lot of these that are compatible with different racks because, as you imagine, there isn't exactly a standard in the industry. Now, if one of our clients doesn't have a compatible rack, then we give them one of these standalone fixtures that essentially is 4 pegs coming out, and it simulates what a slot in a rack would be, right? So then you can simply put your dome into the standalone stand, and then it acts as if it was just in that rack.
So when I walk into a facility, how many racks might I be able, would I see, for example?
Totally depends on the facility. Some can have, one rack which holds 100 cages, some can have 10 that hold 1,000.
Okay.
It depends. Like the scale is something that usually gets limited by labor.
Okay.
Right?
Okay, cool.
Yes, so that's the stand. That's how the dome sits in it if they don't have a cage or a rack that is compatible with one of our domes. And then I also brought our food hopper here,
Okay.
which is really just something that sits on top of a cage.
Okay.
So in this cage over here is just something that a lot of people use, normal cage bottom. Mice run around in it. You usually have mouse bedding in it.
Is this for, what is this kind of plug here for?
Yeah, that plug at the back is where you would connect to the automatic water dispensing system of a rack, which in this case, there is none. That's why you would probably have a water bottle on top.
Okay.
So when you put this in, now you have our food hopper sitting on top of the cage, and it's with a filter inside of it as well. Mm-hmm. And it's very simple to just slide it into your stand, right? Just like you would in a rack. Okay. So you easy peasy, slide it in, and you're done. Okay. Right? So you can remove it as many times as you want, put it in as many times as you want, and the data is always streaming. It knows when you've removed the cage. It knows when there are no mice in it. It does everything for you. So maintenance is not something that you have to worry about. Right.
So can you tell us, one of the things that stands out when I look at the dome, there's this kind of like hardware component here. Can I move this as well? Yeah. On the back end, I see sort of what looks like maybe, I don't know, a fan or and a USB-C port. Correct. Can you tell us more about what's going on in these sort of units here?
Yes. So the fan is there to keep things cool and also provide more ventilation, right? Because you don't want to have mice overheat or just you want to circulate the air. Okay. So that's where the fan plays a big part of. And then these ones are here just to keep the cage. Basically, there's little brackets on the side that keep the cage in the correct position, such that the camera is always looking more or less at the same area. Okay. There's obviously some tolerance in terms of a few pixels between cages, but we make sure things are consistent. So this way, if you perform an experiment with 4 of our domes, and then you perform the same kind of experiment with 4 different domes, the data that will be produced should be very much so identical, assuming the environment and the subjects are unchanged.
Okay, and that's a big deal in reproducibility. Absolutely. Specifically, where folks want reproducible studies.
Exactly.
So that other labs, other research centers can take advantage of the same data. Okay,
yes.
Cool. And then sorry, and in here you mentioned there's a camera system?
Correct.
Okay. Yeah, can you tell us a bit more about that?
Of course. So there's a camera system in there. There are some other sensors, a microphone and whatnot. One of the important things in here are actually LEDs, right? So we have LEDs that do not interfere with the mice, because we don't want to mess up their circadian rhythm or stress them additionally from just having a light shine in their eyes, right? So it's something they can't see, but it's something that we can use to see them. Okay. Right, because without proper lighting, it's very difficult to have a consistent camera system.
Okay, okay, very cool. And then finally, you have your Wi-Fi box, and you mentioned this can, how would I say this? This can communicate with many of these systems at the same time.
Exactly, right? Exactly. Yes, this is just our funnel, right? We take all of our domes, they communicate through this little Wi-Fi box, and you can log in on it normally, just like a normal computer. There's a little GUI or graphical user interface that makes it really, really easy to connect to the network, regardless what the network is. So you plug it in, use the interface, it connects to the network, and now all of a sudden, all the devices are connected, and everybody's happy.
Okay, cool. So as somebody who's working in hardware, what was like an unexpectedly maybe challenging part of this process, or what are you especially proud of in what you've come up with?
Yeah, totally. So in terms of what was challenging, I think the scaling of the hardware is always a challenge, right? Like building something to work for 1 unit is a lot more different than building it to work for 10,000 units. Yeah. So that's a whole problem that I'm sure everybody in the hardware industry is familiar with. One big challenge that we had at Olden was actually when we first started, we pivoted very early on. So our first MVP was to actually build a fully encompassing smart cage where we retrofitted the cage bottom as well. So we had our own cage bottom that was integrated with the cage top, and it wasn't very compatible with a lot of systems out there, right? It was kind of just like our own standalone system. It had more capabilities and features, but it would have taken us a lot longer to actually build and start selling these, right? And as an early startup, you want to get something out there in the market ASAP, You want to prove product market fit. Do people actually want this? Because people can say like, oh yeah, I'd love to buy 1,000 of these. That's great, but until they buy 1,000 of that, it doesn't really mean anything to me, right? Yeah. So that was a way that we could quickly see how people really feel about it, right? So that was one aspect of moving quickly, changing our MVP to, have a simpler solution that was able to provide the same kinds of results, same output, really, right? You still have data analytics, you still have live streaming, and now it's a lot more easy to make, to design, to troubleshoot, and you have something that you can sell for also a smaller price point because it costs less to make it, which reduces that brick wall to essentially one of those very, very, very artsy things that people have in their houses. Like you walk into like a hippie house and you walk through a door and then jingles everywhere. So that's our barrier. You just walk through it.
Okay. Yeah. So it's a jingly barrier to entry, versus a brick wall. All right.
Yes. So that was one thing we pivoted with very quickly, and really, really good decision in hindsight, because we were able to get this thing into customers' hands in less than 6 months from the day of making that decision, versus taking it probably 14 months if we did the full smart cage initially, right? And this is still a smart cage. It still analyzes your data. It still does mouse tracking. It's just a more clever system that can integrate more easily with a lot of other people.
Awesome. And one of the things that you told me is that this is currently being manufactured here in the United States. Is that right?
That's right. The beautiful land of the free.
Okay. All right. Which is very cool. I think there's a big push in, generally speaking, bringing manufacturing back to the U.S. And here we have a startup that's doing that. Cool. Great. Well, so that is the hardware end of things, which is really neat. A lot of the magic, I think, happens on the software end. You sent me over a couple of slides that show your kind of back-end interface and some of the data that you capture. So would you be up for walking me through that as well?
Absolutely. Let's pop it up.
Okay. Cool.
Great.
So I'll let you drive. Yeah,
sure. So this is a very quick demo of what our web app pretty much looks like, what a customer would be seeing, right? So they log into our website and they log into their account, and then they will see something like this, if it loads.
Yeah, this is on me. Oh, there we go. All right. The beauty of... Okay, cool. This should work now.
Great. Yeah, so they see something like this, a dashboard with all of your studies, and one study could be, maybe you're testing how a high-fat diet alters the behavior of a mouse, right? Or maybe you're testing how, I don't know, knocking out some genome in this mouse model alters their behavior and their lifestyle, whatever, right? So you have all these different studies, and if you go into one of them, or before you go into one of them, you can see this video dashboard, and you can see all your animals, all the live streams. And of course, this is a screenshot, but these are all essentially active videos. So on the website, everything is live streaming. And when I say live streaming, it's like within 15 seconds or so. So it's pretty live. Yeah. And this is very beneficial for a lot of animal researchers because they don't have to continuously be checking on in person on all the animals, right? They can quickly pull up the UI and be like, okay, cool, I see animals look good here, 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 here. Awesome, I'm happy with it, right? So that's one thing they can do with their visual eye. But of course, we also have a lot of alerts and statistics that we provide them and actually notify them of, right?
So typically they would be, if they didn't have the live stream, they'd be like pulling each cage.
Exactly. Typically they'd be manually there. Pulling each cage is not an uncommon thing to do. Yeah. Sometimes they have transparent cages, or they have enough of space to kind of look around it. But a lot of the time they have to physically move the mice around. And not only is this time consuming for the scientist, but also it's stressful for the animals, right? You don't want to be modifying the environment of the animals. You don't want to be adding these variables to your studies, right? Because you're adulterating your studies, and it's just something that's been accepted in the industry, which is not okay, and we're trying to change that.
Interesting. How many animals or like mice would I see in a specific cage?
So we support up to 5 mice per cage right now, just based on how much space there is in a cage, right?
Do these cages take other animals as well, like rats, for example, or no?
Right. So our technology can be applied to other animals, but right now we're only focusing on mice. We're trying to keep this product high quality. Sure. And when we're at a point where we grow the team and we can focus on animals like rats, which are a very hot topic in neuroscience, since rats are a lot smarter than mice, then that's what we're going to do.
Wow. Didn't realize rats were that much smarter. The more you know.
Yeah, yeah. Cool. So that's kind of the visualization of all the video streams. And then if you click into one of these cages, you can see there's a screenshot. A mouse is moving in it, hence it's pretty blurry. But that's okay. We can capture all of this. And you can go to the video history, which I'll show you what it kind of looks like in the next slide. But you also see all these beautiful alerts on the side, or the state of the alerts, right? So it tells you if there is enough food in the cage. It tells you if there actually is a cage in the rack, meaning, if a scientist is maybe changing the bedding, maybe they're modifying their experiment, our analysis knows all about this, right? And it tracks it for them as well, so they can also have data points of how many times did they, interfere with the study or adjust it. So it's all recorded, and they can use it for later. We also check for things like, sluggish behavior, or if there are dead mice, right? Because if a mouse dies, you kind of want to know about it right away. You don't want to wait until a day or two passes and maybe it was some weird infection and spread, or maybe it just gonna stress out the other mice or whatever, right? There's all these variables that are introduced, and on top of that, you kind of want to address this problem as soon as possible, right? You don't want to wait. A lot of things can happen between time of death and when an autopsy is being done. So you want to kind of get that as soon as possible to get the best results.
Interesting. I'm imagining that as you're tracking these kinds of metrics around like sluggishness, you might even be able to start predicting, for example, that if we don't take care of this now and have an intervention, this mouse might die.
Yeah, exactly. That's something that's in the pipeline.
Okay.
Yes, and another alert that we're already using is flooding. Believe it or not, flooding is something that is more common than you'd think.
Flooding, like water flooding?
Water flooding, exactly.
Huh, okay.
And that's mostly when it's in a cage, it's connected to this automatic water system, and there's a bit of a leak, or the pressure difference isn't quite there, and then now it's always kind of slightly dripping water into the cage.
I see.
Which over the span of a few hours, it can flood the cage, and
so sad.
if you don't check on the mice for like a few days, they can't swim forever. Right. So there's an alert that tells you like, hey, it's flooded.
Yeah.
Take care of your cage.
Okay.
Right. And it tells you that it's flooded before the mice have to even start swimming.
Okay.
Right. So it can tell if it's wet in there, and that's something that's pretty tricky to do. But thankfully, we have machine learning to do all this hard lifting for us. So there's all these different alerts.
When you say wet, is this happening through just like visual information?
Yes. This is all visual.
I'm imagining as you're looking at all the different variables that can affect the study, for example, I don't know, humidity, for example, could be one of those, or temperature and so forth. Are those currently tracked like by the facility? Is it cage tracked? Is it in the pipeline?
Great question. Temperature to be cage specific, that's in the pipeline. And humidity and and facility wise, that's all being done on the facility level. Okay. There are obviously standards that you have to keep the temperature at a certain, within a certain range for animals to live comfortably. So that's why a lot of vivariums have like a vivarium-wide temperature gauge.
I see.
Yes.
I see. Cool.
Yes. So those are the alerts. Now, if you look at some history, you can see some of the overlays, which are essentially bounding boxes that show you like a snapshot of kind of what our machine learning models see for the mouse tracking. Right. So you can see this for every single frame that has been analyzed, which once again is being done basically live. Okay. Right. So within a few minutes, you have all the analysis. You get your metrics, you have your overlays, and you can select whatever you want from the video history. You can see what happened yesterday, a week ago, as far back as 2 years, if that's when you started your study.
Okay. So one of the names that you mentioned on your website as a competitor is this, I think it's called DeepLabCut Lab, which also does visual tracking. What is like different between your solution and that solution? Yeah,
that's a great question. So DeepLabCut, I wouldn't say is essentially a competitor. They're an open source software that's used for essentially mouse tracking or animal tracking and to analyze their behaviors. Okay. It's a very powerful software, but it's also very, very expensive. And what I mean by expensive is it's computationally expensive. Okay. So for example, if I want to take one of our videos that we record with our hardware camera system and analyze it with DeepLabCut, to get it to analyze it live, meaning let's say you have a 1-minute video segment and you want to analyze it in less than 1 minute, right? That would essentially translate to live.
Yeah.
That costs about $1,000 per cage per month.
Oh my God.
Yeah.
Okay.
So it's not exactly a scalable solution, right? It's good if you want to maybe analyze a few hours worth of data or something very quick, but to get something to analyze it 24/7 reliably and affordably, it doesn't cut it.
Interesting. Wow. So that gets back to that kind of accessibility and scalability question.
Exactly.
Right? Okay. So you have, would you say, is it better algorithmic efficiency than an organization?
Yeah, that's one word I would use for it.
Okay.
The true word is magic sauce.
Okay.
It's part of our magic sauce, but yes, it's a highly optimized algorithm using our highly optimized machine learning models.
Okay.
That essentially gets us to do it for way cheaper, right? I mean, what we're offering this at, and when I say offering, I mean it costs us obviously less to run this.
Sure.
Is less than $50 a month per cage, right?
Okay.
So that's less than 20x.
Interesting. And how many animals can I track with that?
Yep, as many as there are in the cage. So right now, 5 is the limit for these kinds of cages.
Sure.
So that's how much we can do.
Cool. Do you have more slides, or is that the last one?
Yeah, so there's just to show what some of the metrics look like. But first, I'll show you, a quick video.
Oh yeah.
So you can just see mice moving around, doing their thing, and it's a quick 10-second video of what you would see in the web app. Of course, in the web app, this is continuous. It's not 10 seconds.
Sure.
So you can see basically live behavior, and with that kind of resolution and quality and detail.
Wow. What is the typical length of a study, and what is the typical lifespan of a mouse?
Totally. So the lifespan, I mean, if a mouse gets to 2 years, then you're an elder. You're a wise mouse.
Okay.
So yeah, that's not really quite there. In terms of a study, it depends on the branch, right? Neuroscience, you might be looking at a week, maybe 2 weeks, depending on what you're doing. For longevity, obviously, it's in the months, right? It really depends on what part of biology you're studying. So short answer is anywhere between a few hours to the whole lifespan of the mouse.
Okay. Wow. All right.
Yes. So if you look at the next slide, it's just a screenshot of some of the data visualization in our web app. So you can see some of the different metrics that we track, such as max movement speed, or angular velocity, or how much they're climbing, or how much they're traveling in general, how much they're fighting, like aggression, all these different metrics that are nicely laid out and have alternative views. Like you can look at them as bar graphs or scatter plots, whatever you want.
Okay. As a non-scientist myself, I don't have a great sense of what are the standard types of measurements that one takes when they do these kinds of studies.
Yes.
I'm curious, to what extent, in the Venn diagram of things that folks typically look out for and what you're currently tracking, how much overlap is there? And are there areas that maybe weren't previously tracked that you're now able to kind of bring as new data sort of sources to sciences?
Absolutely. So currently in the industry, we use these things called manual assays, which, for example, if you want to test how strong a mouse is after some experiment, you hold out this metal mesh, and the mouse literally hangs on to the mesh. And then you time how long they can hang on for. And based on this measurement, you say, okay, I guess that's how strong they are. Cool, moving on.
Yeah.
So now imagine doing this for thousands of mice. And somebody is just sitting there waiting for the mouse to lose their grip.
Sure.
So those are the kinds of experiments that are being done right now. It's very, very manual, very tedious. And what we're providing here is a slightly different method. So it's in terms of the Venn diagram, there's absolutely strong overlap. Like we can do object association, see how mice are attracted to various objects, which is very important in neuroscience. We can see how much time they're spending with the objects, how much time they're spending with each other, which is social behaviors, how much time they're spending in corners of cages, or how active they are, right? We have all these metrics we currently analyze that are actually more powerful than a lot of these manual assays. So it really is augmenting the kind of data you can get.
Okay, interesting.
Yes.
All right.
And then, of course, those graphs and visualizations are nice and pretty, but it doesn't do much for scientists other than be like, oh, okay, cool. Okay, here's what the data looks like. Nice. Yeah. You actually want the raw data, right? So we have download tool where you can download whatever data you want in terms of the metrics and the mouse tracking coordinates, everything that we provide, right? So we have coordinates for the body of the mouse itself, the head of the mouse, how much they're eating, drinking, sleeping, moving around. And this is running at essentially 10 frames per second. So you have very fine resolution of 100 milliseconds, right? So you have all this data. You can download it for the duration of the entire study. You can also download the associated videos with it. So it's a lot of powerful tools that the research scientists can use to make whatever predictions they want, to make whatever conclusions based on all this raw data, right? And if it's too much for you, well, don't worry. We provide you the metrics that are already calculated. So there are different levels that people can use, right? You can dive very deep into our tools, or you can use it shallow level, right? So how does all this tie in together? Well, like this high-fat diet study that I mentioned, you kind of want to know how the behaviors have changed, right? Okay. So here's one quick analysis that we did in-house.
Okay, so in this bar chart, we have in purple a regular diet, and then in green a high-fat diet. And we're seeing changes in things like angular velocity, climb time, drinking time, eating time, and so forth.
Exactly.
And how those moved. Okay.
And based off of these, you can see what is statistically significant. Huh. And you can make the conclusions that, yes, these things change in this kind of diet. And it's very simple to publish a paper once you have this data and these deltas.
Wow. And so how many, and I know you're on the robotics side, so maybe you don't know the answer to this yourself, but I'm curious, how many mice would, or cages would be involved in a study like this?
Yeah, as many as you want. For example, in this one, I believe we used 10 cages. Okay. So it was, I think, 40-ish mice, if I remember correctly. But you can go use as many as you want, right? Okay. It depends how statistically strong or what the power you want to be is, right? Because obviously larger sample size, more effective your studies, etc.
Okay, very cool.
Yeah, so that's pretty much the whole quick spiel of the web app that I thought was worth sharing in this short session. But there's a lot more that we offer.
Okay, cool. Thank you for that walkthrough. I'm going to pull up my questions here. I'm curious, as you've gone through this process, you mentioned that big first pivot that you did. As you've put this in the hands of the consumers or partners, is there anything in particular, whether it's about the hardware design or the software design, that sort of came from consumer feedback, or maybe a challenge that you've heard consistently that you're really excited to be working on? Insofar as you can share. I know a lot of this is proprietary, but does anything come to mind?
Yeah, absolutely. I mean, I think of the typical tirade of Silicon Valley, where they say for startups, get your product in the hands of customers and work quickly. And you hear this over and over and over again, and you just roll your eyes. It becomes kind of a nuisance to you. But it's the truth, right? You want to get feedback as quickly as you can from these customers. And it's especially effective on the web app and software side, because you might think you know what people want to see, but that's not true. You know what you want to see. You know what your coworkers want to see. People care about different things. You always have this, I think this is common for engineers, you have this bias. You have this engineering bias. Sometimes you have an expert bias. Something that's obvious to you is not obvious to other people, right? For example, let's say when I was first making a study on this website, to me it was very obvious because I knew where everything was. But then when we gave it to one of our first few beta customers, they were like, where do we go from here? How do we use this? And I was like, oh, they're right. It's not as intuitive as I thought, right? So getting that quick iteration, that quick feedback where we can, improve our product on a daily basis while making their lives better, right? We're basically confirming to their feature requests if it's reasonable, and it usually is. We're making tweaks here and there to make it more intuitive, like I mentioned. It's just to make the whole ecosystem more usable. Right, so I that's invaluable. Working with the early beta testers has been really, really invaluable. And for any startup out there, get your product out there in the hands of customers and make sure you have a plan stating what it is they will tell you, what it is that they will come back with in terms of feedback. One of the early mistakes we made was we just kind of gave our beta customers some of our product, and we're like, okay, we're waiting for feedback. And it's like, well, no feedback's gonna come. They're just using it. They're doing their thing.
Yeah.
But like, unless you explicitly say like, okay, here's the plan. We're gonna give you this, and in return, we want to work with you. We want to make this better together, and here's how we're gonna structure it. Once you get that agreement in place, it really propels your product forward.
So all those, accelerators and startup coaches and entrepreneurs are right. Either build or be talking to your customers, right?
They're onto something.
Yeah. Okay. Darn it. Cool. So then we've touched on this in a couple different ways, but taking it back home to this idea of accelerating scientific discovery. I'm curious, how would you summarize how the work that you're doing here on both the hardware end and the software end is really helping us sort of move the needle on that? And especially I think one of the gaps you said is so much of kind of modern R&D is increasingly data-driven, whereas these kinds of animal studies are sort of still kind of left in the analog days. Yeah. So to what extent also can this help us move sort of the needle into these kinds of studies being more, again, data-driven, computational? And again, we've seen an example of that, but curious to hear your own words.
Sure. So I think one of the big things we're trying to accomplish is having an unofficial standard, meaning we want to have data be the norm. We don't want it to be this fun, creative tool that people are using and saying, oh, look at my cool experiment. No, we want this cool experiment to be the norm. Right? We want people to be data-driven, as you said. We want them to care about their animals, follow the 3R principles, which our tool allows you to follow it better. We want you to have reliable and repeatable data, because if it's not repeatable, then we have to do 7 of the same experiments, right, to hopefully find 2 or 3 that are similar enough, right? Like, there's a lot of waste, and waste doesn't just translate to dead mice. It also translates to a waste of time. And time is, as you know, one of the things we can't really get back. So I want to make sure that all the researchers and scientists are focusing on the research and not on the maintenance, the animal worries, remembering if they have enough food, like checking on them constantly to make sure that they have what they need to run the experiment correctly and with minimal variability, right? We want to create this infrastructure that is, like I said, just universal, right? And the tricky part with doing this is getting it in the hands of enough people such that it becomes universal, that they see the value, that they talk about the value, and that everybody can see that, oh, maybe there was a better way of doing animal research, and here it is.
Interesting. So what I'm hearing is a lot of building a platform and creating standards that then help sort of scientists get out of their own way, where they don't have to be focused on a lot of these kind of tedious type processes, and be able to focus on sort of the most value add type research and maximize their use of time.
Exactly.
Okay. In your mission statement page, there's sort of some what seems like longer term language around it says, we're dedicated to building the tools and technologies that enable animal testing less invasively with higher quality data with fewer animals, as you said, using robotics, AI, and genetic engineering. Could you share more, especially about that last piece and genetic engineering and how this all ties in?
Yeah, so the smart cages are the first step to creating a complete ecosystem. Robotics come in to actually automate the lab, which is where our cloud lab comes in, right? So imagine you want to run an experiment, and instead of having to set everything up yourself, you just set up the experiment, and then you say, hey, Olden, can you run this for me? We say, sure. We click a button, or maybe they can even click that button, and now the lab does everything automatically. It changes all the cages, it cleans things up, it replaces food, bottle solution, analyzes everything, whatever you need, right? It's all encompassing. It makes it very easy to actually progress things forward. And the scalability of this is pretty much limitless, right? You just open up more facilities that can do more of this in parallel. So that's the cloud lab. Now there's the genetic engineering that you mentioned, which is where Michael's research from his PhD comes in. And it's you got to talk to him about this a little more, but it's a very cool method of essentially targeting organs, right? So it's like a multi-AAV injection technology. You can look it up. It's from Harvard. But yeah, there's basically a lot that can be done once the tools are in place.
Interesting. Okay, so there's some really cool futuristic genetic research that can be very targeted, that can be done.
Correct.
Ultimately this platform helps enable.
Absolutely.
I see. Very cool. Well, in closing, you're a startup. You're doing some really interesting work. Thank you for shining a light on, I think, a part of the life science research process that I myself didn't know a lot about. Maybe it's not the sexy part of the science research process, but clearly one that's really important. And you're bringing into the modern age in ways that, what I'm hearing from you is also about doing sort of, I guess, the necessary work with animals, but doing that in a way that is also more sustainable, more respectful, honoring them as we do the spiders that we find around our house. I'm curious, where do you need help right now as a company, as an organization? What's one way in which the community folks who are listening could be supportive of your efforts?
Yeah, so at Olden, one thing that we've done well is keep a long runway. So we have, ample cash, we're very lean, we get the work done, right? But the downside of being lean is that you don't spend money on marketing, which means basically all of our clients have heard about us from other people, right? So we're basically relying on the word of mouth effect. So one very powerful tool for us would be to get our name out there, have researchers and scientists actually learn about us a bit more and see if we can help them, if we can help them make their lives better, if we can help them make their studies more robust, right? So that's one massive thing. And the other one is, we are hiring a scientist. We have a lot of animals in our facility. We are always creating new methods, new metrics that can be used for various experiments. Like I said, in the neuroscience space, to replace various manual assays with automatic features. And a scientist is needed for this, not just to take care of the animals, but to actually be creative and come up with these methods. So if you have any great scientists that are looking to work at a startup, let me know. Okay, awesome. So if you're a scientist, you're inspired by this vision, look them up, Olden Labs. Oldenlabs.com, or you can email us at info@oldenlabs.com. Beautiful. Michael, thank you so much for your time today. Hope we can have you on again in the future, and good luck to you. Thank you, Nabil. Great being here. Cheers. Take care.